title is Exploratory Learning in Biological Cells, and what I'm going to do is I want to talk about one particular type of learning. You guys have been thinking, I think basal cognition is the title of generalized learning in different embodiments. One particular type of learning that I've been interested in is exploratory learning. And I wanna, this has roots in psychology and in neuroscience. I don't have to yell, right?
No.
It feels like you don't have a microphone, so you.
You can hear, okay, right?
I'm talking too loud.
Yeah, okay, you can hear.
All right. So this particular kind of learning has roots in psychology and in neuroscience, and I'm going to present it in this context first. And then I'm going to talk about how we kind of transfer this or apply it to cell biology in the context of gene regulatory networks. This is quite old work by now, and then I'm going to talk, time permitting, about our efforts following that to model this type of learning in random networks. And the bottom line of these modeling studies can be stated in a way that also resonates with some of this group's result, is that learning is feasible, but it's not universal. It doesn't happen with any just random networks. It depends on certain properties. But on the other hand, I will argue that these properties are not so rare, and they're shared by many real systems. So this just might be a relevant type of learning for cell biology. Okay, so I start more than 100 years ago with Thorndike's Animal Behavior, a classic book in animal behavior with the same title. And I want to refer to one of his experiments where he took cats. Okay, he had built these puzzle cages, and he placed hungry cats inside the puzzle cages, and they were supposed to find their way outside to get to the food. And the cage was built such that there was some lever or something to be pulled or pressed, and the animal had to find that in order to get out and get the food. And then he repeated this. After the animal did that, it was repeated many cats. And what he found was that first, for a while, these animals just tried out many different random motions, many different ways to move their body and their limbs. And at some point, they hit upon something successful that brought them outside to the food. And his observation was that over long time scales, after many repetitions, they had associated this last successful motion with the reward. And so then they could repeat that much faster. And what you can see here, this blue line below, cursor a little bit more here. So what you can see here below the box is a graph of the average time required, the escape time, how much time it took the cat to get out of the box as a function of trials, and you can see that there's a decrease. So this is a type of learning. The cat has associated the successful actions with the reward. And so, so this is the behavior that Thorndike formulated as behavior leading to a satisfying situation or avoiding a stressful one are likely to be repeated. This is a kind of trial and error. And many years later, I mean, it's like already 20 years ago, but much later than this original experiment, Shimon Maom, who is a member of our center, decided to take this from the behavioral level down several levels of organization to a ex vivo network. And I heard already this morning that people are working with or have been working with such networks, so maybe you know this already. By the way, I didn't say this, but please stop me if you want, if I'm saying obvious things or if you want to ask. So here's Shimon's experiment. There are neurons taken from newborn, usually rats, and placed on this chip with electrodes. And you can talk with this system through stimulation and recording in any one of the electrodes. Hananel can say about the system much more than I can. But here, the thing I want to point to is this experiment design that was formulated in order to put to the test the question whether such a system of neural substrate, which has no anatomy and is not embodied in an animal, can perform in a way similar to drive reduction as formulated by Brenner. So here's the translation. There's the task that was defined for the system is a specific input-output response. So I choose one electrode as the stimulating electrode, and then I require the system to respond in another predefined electrode. And the assumption which has been studied beforehand is that electrical stimulation drives the system to be modified, to modify its connections, to modify its state as a network. And so when you put in stimulation, you're driving it to change. And so the idea was that I'm just driving. Once I get the required response, I shut the driving down. So this is a closed loop system with a computer. And when I read the response that I had anticipated or I had required, I stop the stimulation. Okay, so this is the analogy of the cat's hunger that's driving it, and then when he's already not hungry, he's not searching. All right, so here's an example of a measurement. So this is one electrode. So if I choose this electrode to answer, to respond, after a predefined time window, 50 milliseconds in this case, a priori, before the experiment, there's not a lot of action in this time, not a lot of response. But after the experiment is finished, I have a lot of response at this time window. Okay? And the fact that this experiment actually worked is a highly non-trivial result. Okay? This is an implementation, again, in an ex vivo system of this animal learning or animal behavior concept. So here's what the escape times look like. I have on the x-axis the trial number, like in cats, and I have in the y-axis the time to the response, to the required response. And these are jumping up and down. Also the cats do this, also animal behavior does this. And when you average over many trials, you get this decrease, a smooth decrease in the response time. Response time is the time that it takes to get the required response, and then you will say the system has learned what I wanted to teach it. And this is the control. If you open up the loop and there's no response dependent on the system, then this time just increases. All right, so does everybody know this paper? Or no? Is it clear though? Yeah, okay, so I think this is a highly influential paper, although many people tried to repeat this experiment with not a lot of success. So experimentally, this has not turned into a paradigm in neuroscience for reasons that I don't really understand, but I think conceptually it was very influential. So let me zoom out for a second and just say things that I think in this group are going to be quite obvious to all of you, and just kind of make a contrast between two ways of performing a task or inducing a desired behavior. One of them is a deterministic algorithm, a series of directed actions. For example, I could write for you if the cat could read, I could write for the cat, do this, and after that, do that, and then give them a series of things. This has analogs in other areas. For example, when you think about learning, what's the algorithm for strengthening the synapse, right? There are time-dependent plasticity rules. So there are microscopic rules. Same with gene circuits. When this gene is up, that one turns on. This is could be an efficient way, but it's very specific. It has microscopic, well-defined goals, and it's repeatable on all levels. And I will call this an instructive way of performing a task. The problem, of course, is that this is limited. You can only store so much information about a finite list of tasks that you can work on in this way. And to contrast that is the other extreme is trial and error or exploratory learning, where you do something, you try something out, you estimate your error from some feedback, and then you modulate maybe through a random walk. And this type is inefficient, but could be inefficient, but it's very, very general. I mean, if this works like we think it does for the neural network and for the cat, then in principle they can learn anything. And an important feature of this is that there is a macroscopic goal, but I do not instruct the system on a microscopic level how to do this. And so there could in principle be multiple solutions, okay? And this somehow is a selective way that I would like to fortify or strengthen the correct or stabilize the correct trials. Okay, so what we tried to do then at that point is, this is a joint work with Eris Brown at the Technion. We wanted to take these concepts and apply them to cells, okay? So we're going even one step below the network that was composed of live cells and all the other things, glia, etc. And we're going to the single cell. And then we were thinking how this relates to gene regulatory network. So gene regulation is often thought of as a control system that has the first type, the instructive type of structure, right? It has, you can make a map of the interactions. This gene drives that gene, etc. And there's a blueprint for a program. In this case, no, this is the developmental program for a sea urchin. But actually, gene regulatory networks do much more than this. And if you look throughout evolution, gene regulatory networks are responsible for a lot of the phenotypic innovation that you see throughout evolution. And this has been studied by observing the animal kingdom, by observing the natural world, and is known under the name of co-option or gene recruitment. So what happens is genes sometimes cut and paste themselves in a way that creates novel structures, and then you can trace that. For example, one of the most beautiful examples is with butterfly wings, how the transcription factors mix and match to create the different patterns on the wings. Okay, but this is out in the wild, and our goal was to bring this somehow into a lab setting and to design an experiment that can test whether single cells can in fact learn through exploratory learning or through something which is analogous to this. Okay, so when you mix and match or cut and paste a regulatory system like this, something that is structured, in principle, nothing, it cannot revert to the typical or the hardwired recipes that it has. It has to do something different in order to overcome these perturbations and to come up with something. So here's the experiment that we thought about. We took yeast cells, single-celled organisms, and we kind of did a gene recruitment synthetically in their network. So yeast is great because we know a lot about its regulatory system. You take a metabolic gene, which is essential for growth, and instead of it being driven by a regulatory system that reads the correct inputs and responds like a normal input-output system, you completely fool it. You kind of connect it to something completely unrelated to the function of the gene. And this is a gene that if you knock it out completely, the organism is going to die. But if you connect it to a foreign regulatory system, or you can, for example, shut it down completely by external signals, that creates a frustration in the system. But interestingly, in that case, the organism does not die. So here's what happens. These are normal yeast cells. You plate them on a Petri dish, and after 2 days, you get these many little colonies, right? This is how yeast cells grow. But after you do this perturbation and rewiring of the gene regulatory network, after 2 days, you see nothing. So you might think that these organisms are just going to give up and not work their way through this conflict. But if you're patient, then after 14 days, which is an enormous amount of time, you start seeing the colonies grow. Okay, so they look very different than the normal colonies. It's a dynamic process. It takes a long and variable time. Every colony pops up at a different time. But on the other hand, many cells adapt independently. It's not a Darwinian process. It takes place on the plate. You can see that between 50 and 70% of them actually find a way around the challenge to go. Do you have a question?
Yeah, this may be very random, but has anyone looked at P53 in these?
What do you mean looked at?
Yeah, so there's stuff with like cavefish, where basically if you put them in a stressful environment, they will downregulate, the stress kind of swamps the protein folding system.
Yeah.
And in particular, that's been kind of linked to P53. Some work from Susan Lindquist and David. So I was just wondering, it could be similar.
Okay, so this experiment has been repeated several times with different types of rewirings.
Cool.
I'm getting to this, maybe this is gonna answer partly. So this is a physiological description of what they do. Now, if you go into gene expression, right, start measuring expression of particular genes, then surprisingly, despite the fact that the perturbation or the rewiring was very local, you take one gene and connect it to a different place, it's a huge response. Hundreds of genes go up and down in response to this challenge. It is stress sensitive. If we have an experimental knob that turns the stress up, and then the gene expression response becomes larger, even larger, and it's non-repeatable, which is maybe the most important feature. So what does it mean non-repeatable? I have here two experiments. This is already old stuff. We measured it with microarrays, like I don't remember how many years ago, more than 10. And so what you're seeing here is the log fold change in genes. Every point is one gene. There's 6,400 genes in the yeast. And some of them go up, some of them go down. But the interesting thing is the next day, you do the experiment again, experiment 2, and you see that those genes that were highly upregulated in the second experiment can be downregulated. There's no correlation, right? This big cloud here says that every gene has more or less randomly gone up and down. I cannot find any particular correlation with what they did in the last experiment. But still, both of the experiment on a physiological level look very similar. Okay, so this tells us that there might be here multiple solutions to the same challenge, multiple solutions on the microscopic level to the same physiological challenge. Okay, and the thing about, you asked about P53. So at some point after we had these measurements, we said, okay, let's look for genes that we know that are related to something. So in particular, the yeast cells have a very well-known universal stress response module that lights up in certain cases. And although we were really desperate to look to find, there was nothing to be found. And not only that, but the modules that people have mapped for yeast are completely broken down in this experiment. New correlations are built and typical correlations are broken. So it's a completely different type of experiment. All right. And I can just say that this experiment was, at the time, people had a very hard time accepting these results. And Ers Brown, with other collaborators from Germany, took very long time and other experiments to, for example, convince people that this is not Darwinian evolution. But more recently, this has been reproduced and extended to different types of promoters and different types of rewirings in another lab, and the qualitatively the results repeated very much. Okay, so could it be that what we're seeing here is a form of learning? This is the question, right? So we have a complex system, the gene regulatory network, right? It's complex, it's multi-dimensional. And not only that, we know that gene regulation is context dependent, right? This has been known from the early days of binding experiments. People have noted that there's context dependence. And context dependence means that connections can change. They have the ability to change. They have that in them. And what could in principle or potentially support such changes, there are many molecular mechanisms. Just naming one is the intrinsically disordered proteins that are recently being rediscovered again and again, and being, years ago it was considered something very esoteric, but now we know that, for example, in the human genomes, 2/3 of the genome has many alternative forms. And not only that, but this property is extremely prevalent in transcription factors, right? Those that are supposed to be the mediators of interactions, those have really a lot of random protein domains. And the one more ingredient, so this is a paper by Niklas. It's called Rethinking. I took the title from his paper, really interesting. Rethinking Regulatory Networks, and he lists a lot of molecular mechanisms that could support the modifiability of gene regulation. And the last thing is we need a global signal, right? We need this something that will serve as the drive. That was the hunger for the cat and the electric stimulation from the computer for this neural network. And we suggest to think about cellular stress, which is something that is sensed by many mechanisms. So there are mechanisms in the cell to sense stress. So this cellular stress could serve as the generator of a drive reduction. Okay? If the cell is feeling or sensing a high stress, it could mount a type of response that is exploratory, and once it is hit on a good configuration, stress would be automatically or spontaneously relieved, and then stability will be conferred. Okay, and this rethinking of gene regulation also has implications for modeling. Again, I go back to this picture, which has become, for me at least, this kind of a prototype for this blueprint modeling of gene regulation. And instead of thinking about mapping out the interactions very specifically, perhaps we should move to something more similar to neuroscience. Computational neuroscience, basically, in neuroscience, you don't think that every neuron has a specific role. So a lot of the modeling is done by random networks. Actually, also for gene regulatory networks, people have done modeling by random networks many years ago already. But things that I would like to highlight is in addition to the random network structure that I'm going to put into the modeling, two important ingredients are, one of them is the plasticity of the connections, right? So now we're kind of getting closer to machine learning, right? I have connections that can change, and they're flexible. And the other thing is the feedback. Feedback is essential here. You want to see how the system behaves under a low-dimensional global feedback signal. So this just might be sufficient to describe something similar to what the yeast cell does. And the great thing about it is that no sophisticated computation is required, right? I don't need to invert matrices like I need to do in algorithms of learning. I don't need to store memory necessarily. And so this might be a type of learning that has its trade-offs. It's very simple. It has little computation, and it could provide some abilities to the cell. Maybe this is a good time to ask if there are any questions till now.
Were these cells cultured together, or could they have learned it from each other, or is the learning going on?
That's a great question. That's a great question. So our measurements were our measurements of gene expression were bulk. We didn't have single cell measurements at the time. I have to say I've been trying, I hope this is going to come out, to have a second generation of this experiment where we can look at single cells, but certainly my feeling is that there is an identity to the population, which means that they are probably exchanging some kind of information with each other. That's a great question. Yeah.
So when you say that the gene expression measurements were in bulk, how did you then produce that plot where you showed like two different experiments having very different?
No, these are two different experiments in the sense of I do the whole thing from the beginning. I take a new population out of the freezer the next week or so, and I start the entire experiment again, and then I get, okay, so the single were those are single genes, not single cells.
Yeah,
I have,
So-
even
so-
with the microarray technology, I have a measurement for every gene, but it's average over the population.
okay.
Does that answer your question?
Yeah, so the first axis is an average of the population of the first experiment.
Yes, and every point is a different gene.
Right.
Okay, but every point has information about many cells.
Okay.
All right, more questions?
So you cannot know the variation of those points.
No, unfortunately, I don't.
It's only average.
It's average over the population, but the variation appears over repetitions of the same, nominally the same experiment.
Right.
You repeat the same protocol, and this is what you get.
Do you think in general there's some kind of intermediary like rules that they follow? Like, yeah, any of the single genes in and of themselves may shift up or down, left or right, whatever the way you think about it. But in general, like there's some kind of like these genes and these genes need to have some kind of relationship that's held.
Yeah.
Yeah.
I think they create new correlations. This is
Okay,
my feeling.
I
I
see.
think I think I'll say more about that later,
Okay,
about the
okay.
relations between the genes.
Okay, that's great.
For the scatter plot again, did you do a control where you had cells that were just happy and not stressed and
Yeah,
compare across
Yeah,
two?
we did that. There's much more. So, okay. So, there's much more correlation when you have well-defined conditions and just normal cells. There's not 100% correlation, but there are those correlation.
And is the cluster tighter, or
Sorry?
is like the cluster tighter? So like in the one you had, it sort of
No.
expanded. Oh, okay.
Yeah, no, when you have no perturbation or no stress in the system, you get this, I didn't put it here, but it's in the paper. You get a much higher, like 85, 0.85 correlation between the same system over time, over experiments. All right, other questions?
I think Navneet had a question on the chat.
Oh,
I
okay.
can't really.
I can't read the chat
Yeah.
because I'm.
Okay, sorry. Yeah, my question is the faster response times of animals was that based on the same animal's self-learning experience or observing other cats?
No, the same animal did the experiment multiple times. Same animal.
Okay, so it's a personal experience, we can say, for exploratory learning. It's not observing other animals
No.
in
In
conformity.
the cat experiment and in the neural network experiment, it was a repetition. So the way they did it is they did this experiment and then waited for a while, both cases, and then did it again. And then there's like a sequence of these experiments.
Okay,
And the decreasing time is across repetitions of the experiments.
okay, makes sense. All right, thank you.
Okay, so what I want to do now is to talk a little bit about our modeling work for this, and I'm going to get into a kind of a shallow, not a great deal of details. If there will be interest, I can give more details later, but I just want to give the general idea first. Okay, so here's the model that we created, and I'll go through this qualitative description of it. I have a network, okay? It's a random network. And I require something from this network, just like the neural network experiment. I require a low-dimensional constraint. This is important. I do not require a specific pattern of its individual elements, but instead I require a low-dimensional projection to be under some constraint that I define, right? So this is what I want to teach. I want to teach the network something that is a low-dimensional task. Now, initially, this network is not going to be in line with the demand, with the constraint. And therefore, my model will return a variable that I will call stress back to the system. And this stress variable, as long as it's high, is going to drive exploration of network connections, right? So imagine, this game where you hide something to the kids and then they go and you say hot, cold, right? So I give the stress when it's far away. The stress depends on the level of, some closeness to the requirement. And the dynamics goes on, and at some point, just like the cat is going to bump on or find a configuration that decreases the stress by complying with this demand. And at that point, automatically stress is going to be relieved, and then the search is going to be stopped and stabilized. This is all in words, I can give equations later. But why this is a non-trivial model is the following reason. In contrast to a two-dimensional room where you hide something and you say hot, cold, and then finally you find whatever it is that you were hiding, we're talking here about a network of order 1,000 dimensions. So there's order 1,000 dimensions squared connections. Now imagine 1,000, or even not squared, just several thousand degrees of freedom, just random walking through space and searching for a configuration. What are the chances that it'll ever find anything meaningful? That's the reason why this is a theoretically interesting problem, right?
The assumption is that the stress function is a gradual, slightly graduate, right?
Right, but I will say more about this later, but the answer is that it's not very sensitive to the way the stress is driven back to the system. It could be a step function, could be a smooth function. It's not extreme. It's a little bit sensitive, but not extremely sensitive to the way you feed back the stress to the system. Okay, now the short answer is that sometimes this converges. And I go through the dynamics, I simulate these equations, and then I would like to ask, what are the chances that this converges to something that I define? And the short answer is that sometimes it converges, and here's an example. This is a network of size 1,500. This is a network which is kind of stressed, right? It has the red or orange color, it's under stress. This is the projection. The projection defines the stress which feeds back, and here is the dynamics. So the dynamics goes on and on. The projection is also changing as a function of time. And as long as the stress is high, also the J is the connection matrix. So the J, these are just 4 out of, I don't know, whatever number larger than 1,500 I had connections in this one. And they're slowly changing. So there's a separation of time scales here, if you notice, right? The dynamics is fast, the low dimensional projection is a little bit slower. And actually the connections which are being modified, that's the plasticity, is much slower. But it's enough, and it doesn't change by that much, but there are many connections. So eventually at some point, the system finds a good configuration, and at that point, it hits a fixed point, and that fixed point complies with my demand, and then the system goes stable. Yeah, question?
Is it appropriate to think about what you're modeling here is kind of like a large network of homeostats? Kind of like the Ashby homeostat, where
That's a good question.
they're basically random connections that we're allowed to change up or down, some,
Yeah.
whatever.
Yeah.
You put a fixed point and they bounce around and eventually will find their way.
Yeah, I think that
With-
it's a high dimensional.
High dimensional
I mean,
homeostats.
I've read Ashby's
That's exactly
work,
what
and that's in the direction.
So it's not bad to
No,
use
it's
that
not,
as a
it's
thing in
not
my
that.
mind, okay.
Yeah, because the system is kind of self-doing something,
Okay.
self-monitoring, self-modulating until it finds an attract.
Cool.
Okay, now, but what I told you is that, when does this happen? That's the question. And the answer is, I have the answer, yeah. This is the short version of the answer. And the result is that it can work, like in the example that I showed you, but it doesn't always work. And this question, whether it works or not, whether it converges or not, depends on the network structure, right? And the short answer is that outgoing hubs in the network support exploratory learning, right? How do we know this? I'm now getting a little bit deeper into the mathematical part, into the computational part. So this is how we studied this. So we defined a collection of statistical ensembles. So you know that in a network, there are outgoing connections or incoming connections. Now stop me please if there's a question. The network is characterized by some structure, and in statistical terms, we say there's a distribution of outgoing connections and a distribution of incoming connections. And we created this collection where we picked every one of these distributions with a different character. So for example, the simplest network is one where you have a binomial distribution of incoming and outgoing. Is that a question?
No, it's someone get
Coming in or popping
okay,
out, yeah.
okay. This is the simplest network over here at the bottom. You just take 1,000 nodes and then you just, with some probability, you connect any two of them at complete random. This network has not converged ever in our experiments, even for very long times. It can never learn the thing that we tried to teach it, all right? Another type of distribution is a scale-free distribution, which has a long tail, which means that you have some nodes that are really highly connected, right? There's a range, but some of them are really highly connected. And okay, so you can see that different topologies and different structures of network behave very different, from 0 to more than 70% learning capacity. What that means is that if I ran the simulation for 100 times, I get 72 simulations where this network was able to learn. Question?
For the simple network, do you think that the stress is too high so they don't even try it, they can't learn, or is it?
I think I can say a little bit more about that later as this unfolds, okay? All right, so what you can see is that, first of all, the different topologies behave differently, and secondly, the three best ensembles, or the three best types of networks, are those that have scale-free in their outgoing distributions, right? So that means they have nodes that are simultaneously driving many other nodes, okay? All right, why is this important? Let me go back to yeast cells, for example, for a minute, and try to connect this to gene regulatory networks. So we know that people have mapped out, there are more modern papers, but this goes back 20 years, and people have mapped the structure of gene regulatory networks by different experimental techniques. And what you're seeing here is a distribution, experimental distribution from binding experiments, showing what the distribution of connections are. So here's the picture for yeast. Okay, if I'm a gene, I am regulated by a relatively small number of genes, not shown here, but if I'm a transcription factor, right, this is the graph for transcription factors, a lot of them are regulating 1, 3, 10 other genes, but some of them, look at this tail, okay? I have few genes, here's 1, here's 2 genes, I have a few of them that are regulating hundreds of other genes, all right? And these are exactly these outgoing, not scale-free exactly, but I do have these nodes that are affecting simultaneously hundreds of other ones, okay? And if this is only to flash the experimental, I'm not going into the details of the experiment, but the experiment shows that out of this collection of ensembles, the closest one to experiment is this one. Is the one that has a scale-free out distribution that you can see here, and an exponential in distribution, which I'm not showing, okay? So this gives me a hint that in networks that look like gene regulatory networks of the yeast, which are not completely random, they have structure, maybe this algorithm of exploratory learning is indeed feasible. It converges 60% of the time. Okay, also in the experiments, as I showed before, this doesn't work 100% of the time, but it works a significant fraction of the time. Okay? All right, so the picture that is starting to emerge is that maybe what random systems cannot do, still regulatory systems can do, because they have topology, they have structure that supports certain kinds of statistical learning. All right, however, if you're a little bit computationally inclined, then people see this and they jump up and say, this is not a scale-free distribution, right? If you've worked on these avalanches, this is a debate that goes on for decades. there's just like several dozen, 24 points here. Is this a power law? Is this not a power law? But still, I can see at the edge, I can see these large hubs that are affecting hundreds of other genes, right? So my experiment in my model is the following. Take a look at this. I'm going to skip this one, but take a look at this right-hand image. I'm going to start from the network, which is very generic and does not know how to do exploratory learning. And then I'm going to add one by one a number of hubs. And like I said in the beginning, when it's a completely random network, it does not know how to learn at all. By the time I've added 10 out of 1,500, right, 10 outgoing hubs, I get 40% learning. What this comes to tell me is that I don't really need this mathematical structure of a power law. I had used this, when I wanted to create different topologies, because this is how we think about it in statistical terms, but we don't really need that. All we need is a handful of hubs that control many, many other genes. And this, I think nobody will argue, despite the the sparsity of this graph, nobody will argue that there are master regulator genes that regulate many, many hundreds of other genes. So this again is a property of gene regulatory networks that tends to support this exploratory learning. All right. And this is just an illustration from some random paper that studies, biologists, there are many, many studies on master regulator genes. Okay, so how does this come about? How do these hubs, like the small number of hubs even, how do they affect this search process that we saw? And so what you can see here is, okay, maybe I should go back and say one thing. I want to compare this ensemble that I said is really good in learning, right? It has outgoing scale-free distribution and an incoming exponential distribution. And the one next to it has the same pattern except switched, right? It's just like transforming the matrix of connection. Now, if you're a theoretical neuroscientist and you think about eigenvalues of these matrices and a lot of things, a lot of properties are actually completely blind to this transformation, but not this type of learning, not this type of algorithm that we're performing here. You can see that if I switch the outgoing and the incoming, a dramatic change happens here.
It seems, according to this table, it seems that the output is matter, but the input is not.
That's right. It's very
all.
insensitive to the incoming distribution.
Yeah.
The outgoing distribution is the most important. I agree with that. Other remarks? Okay. So what I'm doing now is I'm taking these two ensembles whose matrices of interactions are transformed one with respect to the other, and I'm just running the dynamics without any learning. Okay? So this is the binomial scale-free, which means the outgoing is binomial, the incoming is scale-free. Right? Every gene is the opposite of the gene regulation. It can be affected by many others, but affects just kind of randomly. And what I'm doing is I'm running the dynamics many times and I'm clustering it. This is just a kind of a standard clustering algorithm to the time sequences of the dynamics. And what I want to show here, and you can see it by eye without any fancy analysis, is that these guys are going almost independently. There's not a lot of correlation. So the point is that every node in the network has a different dynamics, and they're not very correlated. On the other hand, this outgoing hub network has groups of genes that are highly correlated. Okay? And it is this one which is successful in learning, and this one that fails. Right? So remember in the beginning, we said, look, if we have 1,000 degrees of freedom and there's many, many connections, and what are the chances of every one of them trying to look for a random configuration and in the end finding something? And now what we see is that those networks that are successful are actually not doing that. They're kind of roaming around in the high dimensional space, but on a lower dimensional length. They're not filling the entire space. And that comes about because of their hub structure. They're still random, they're still, even though they have hubs, they're still pretty high dimensional, but much lower than those that are not. And indeed, you can take from these, now if you want to quantify this, you can compute a participation ratio. Again, if anybody's familiar, this is really standard in neuroscience when you measure many, many neurons and you get a lot of data in high dimensional space. You can estimate, there are many different ways to estimate the dimensionality of the dynamics. This is just one of them. And I'm plotting here the dimensionality. I'm plotting here the participation ratio for these three ensembles that we used in the computational model. And indeed, those that are scale-free something, right? You said that the scale-free out is important. So also for the dimensionality, these three network ensembles have outgoing scale-free and three different incoming, but that doesn't really change the dimensionality. And these guys have exponential outgoing and something or other incoming, and these guys have the binomial distribution for outgoing connections and any one of the three for incoming. And you see that the hierarchy is reflected also in the dimensionality of the dynamics, right? So the answer, the qualitative answer is that the existence of these hubs sort of constrains the dynamics to a low dimensional manifold while it's searching around and doing its exploratory thing to find a new configuration. Okay. Now if I go back, I want to go back to the learning itself. This was just free dynamics. Okay? I looked at the network ensembles without any learning. I just looked at their typical way of performing the dynamics that I put into the model, and I found this relation with dimensionality. Question? Okay. Now if I want to go back to this learning, to exploratory learning, again, I don't want to go into a lot of details. This is a slightly different version, which is a little bit more schematic than the original ensembles, but it still has one particular hub it's a network that has random connections, and in addition to that, it has one hub that controls many of the nodes, and it behaves similar to the scale-free out networks. And within these kind of network ensembles, I can do the following. I can change the strength of the hub. Okay, so if the hub is kind of constraining the dynamics, then I can think maybe the strength of the hub can be a knob to modulate this constraint. And what I find is the following. So take a look at one of these graphs. I have the strength of the hub on one hand, on the x-axis, and I have the convergence fractions, which is the ability of this network to learn, on the y-axis. And what I see here is that there is a trade-off. If the hub is very weak, so in this model, if the hub is 0, I go back to the networks that do not know how to learn at all. Now I gradually increase the strength of this hub, and then I get to a really good 20 or 25% learning ability. But then if I continue to increase the hub, at some point it's too much, and I'm going to decrease the ability of this network to learn. So again, the qualitative conclusion from this is that the biological networks have hubs that control a large fraction of the network, but not too much. If I controlled all of it, then it would not have enough search power. So at this end, there's too high dimensionality and the search is inefficient. On the other hand, on the other edge, there's too low dimensionality. The hub is too strong. It's not letting the system search enough. We call this low expressivity. And somewhere in between, in a very broad kind of maximum region, then there is coordinated efficient search that enables this kind of work. Okay. Questions? Yes.
Just very vaguely, in general, would you relate this to something like edge of chaos type things. If you're too far to the left, everything's essentially random, so the search is just like as you wish. If it's too far to the right, it's so controlled, it's too ordered, so nothing can occur.
Yeah, I mean,
You have to, I know I'm pushing it, but it's
no,
something
I
like
think
in
it's,
between.
it has the flavor of the trade-off at the edge of chaos, I agree.
Cool.
Because over there as well, you say that one side is too not expressive enough, and the other side is too constrained or something like that.
There has
It
to be
has the
something.
same flavor.
Okay.
Yeah.
I'm making sure I attach things that you're showing to things I already know to make sure that I remember.
No, I think you're right. I mean, let me just say that this is unpublished stuff yet and not very, I mean, it's quite preliminary.
Yes.
Okay, the last two slides are a bit preliminary, but this is the intuition. Okay, I think I'm gonna finish with the computational part here. So if there are any other questions, maybe this would be a good time to raise them. And then I'm just gonna go and touch base back to biology for a minute before wrapping up, okay? All right, so, more questions? Okay, let me say something about this role for master regulators. And here's an example. This is an example of a master regulator called ZIB1. Which is known to people who study cancer or epithelial, EMT, epithelial to mesenchymal transitions. It is a repressor of epithelial genes, and it's associated with, developmental processes, of course, but also with aggressive tumor behavior, with metastases, with drug resistance, and it's linked to promoting stemness. This is a really important gene in the study of cancer. And it's a repressor. Surprisingly, quite recently, people have found, as a paper from 2015, that found that by teaming up or interacting with another transcription factor or regulator, this ZIB1 hub completely changes its character. From being a repressor, it changes to being an activator. The details are not important. It interacts with another regulator to control a whole different set of genes from what it was before. And the reason why the details are not important is that if you go through the literature, this is just one case, but there are many other cases like this. And people find master regulators that under certain conditions completely change their character, their function, the other proteins that they interact with, and their functionality. So this is a type of rewiring or reprogramming that these gene regulators are capable of undergoing, which is consistent with this picture of exploratory learning or exploratory dynamics that finds novel types of phenotype. And so what we think is a really powerful tool for the cells is that master regulator plasticity can organize new patterns of gene expressions and express new phenotypes. All right, so follow-ups on this, on our work in the direction of cancer, if anybody's interested. One group did a computational modeling that took actual measurements from tumors and tried to ask whether the gene expression as a function of time during metastasis progression can fit into this framework. Another thing that we did is really we went through a whole lot of literature on cancer, and like I said, we found a lot of things that qualitatively support this picture that it's an important process. And so let me summarize, going back to the dichotomy that I drew in the beginning of the talk of this instructive versus selective way of learning or performing a task. What we think is happening is that gene regulatory networks have this dual way of working. On one hand, there's instructive, of course, obviously, a lot of people have mapped out modules and parts of networks where we can trace the gene 1 activating gene 2, etc., and carrying out a very well concerted function. And people have done a lot of work on that, and they know to understand it and to measure it. And this is all there. It's all this instructive performance happens. But sometimes, under certain conditions, maybe under very strong perturbation, maybe under rewiring or unforeseen challenge or certain stresses, the same structure, the structure of these master regulators now carries a new function in the world of search algorithms, where it can reduce dimensionality and enable a completely different way of responding, where new phenotypes and new configurations can be found. And it is this, this dual type of behavior of gene regulatory networks that makes them different from, from hardwired networks or other artificial networks that we know. Okay, summarize. So with roots in behavior psychology and experiments in ex vivo in neural networks, exploratory learning, I hope I've convinced you that it can be studied both experimentally and theoretically, and gives a lot of challenges to think about. The particular model that we developed is a biased random search in a high dimensional connectivity space, and the outcome is highly variable and sensitive to several details. Not to one-by-one details, but in this case, to statistical properties. And in particular, networks with outgoing hubs do well in this type of learning. And the role of hubs can be mapped onto a feedback system. I didn't talk about this work, it's very technical. And it could be that this is implemented by an effective dimensionality reduction in the search. And going back to the feasibility, then I would claim that both feedback and plasticity and heterogeneous connections are all typical of these gene regulatory networks, possibly other cellular networks as well. And they're not completely generic. They're not random networks like we use in physics or something like that. So let me just acknowledge my main collaborators. There are many have been over the years. So the work, experiments on yeast cells were led by Ayers Brown at the Technion, and these people were very involved. The model was developed with Yoav Swain at the Weizmann Institute, and the main player was Halil Schreier, a PhD student. And with Omri Barak from the Technion, who is a computational neuroscientist, we are working on the general framework of the theory of cellular learning. And in terms of further challenges, I think understanding the master regulators as hubs is really still underway. We don't completely understand it. We have some convincing arguments, but not enough evidence. And what is really interesting to me, and I think in this group it's probably resonating with some of your works, I would really love to understand how these and how similar principles are implemented in other organisms and other levels of organization, other embodiments. From the practical point of view, I think it's a nice challenge to design a specific task to be learned by cells. For example, many people are trying to teach bacteria to metabolize plastic or whatever, and they're doing that mainly by instructive gene biotechnology. But perhaps this can also be done by designing experiments that are in principle behavioral experiments. And other forms of primitive learning is another thing. Yeah. All right, thank you very much for listening.
Great. Anybody else questions?
I do have a question if you can hear me.
Yeah. Sure.
Okay, cool. So the idea that having outgoing hubs and networks will effectively reduce the dimensionality of the system makes a lot of sense. But would it also constrains the number of the variety of problems it can solve? So in your system specifically, it's your fitness function is the low dimensional constraint, right, that you impose. Could it also mean that the number of ways you can formulate that fitness function is also reduced
I
due
think
to
so.
networks with outgoing hubs? Okay.
I think you're right, and I mean, I stated it in some way in the beginning. I think this kind of primitive learning, to me, it's very appealing because you need very little for it. But on the other hand, I agree that it cannot do a lot. There's a limit to what it can do. And I totally agree, yeah.
Okay. Thank you.
Anyone else?
Yeah, so do you have any idea what the mechanism could be for the low dimensional stress detection? How it knows it's far away from homeostasis.
I mean, cells have mechanisms to stress different kinds, to sense different kinds of stress. For example, protein is folding, and for example, temperature, they have these, I don't know, I don't have particular mechanisms, I just kind of rely on the fact that stress causes a lot of things in cells. It triggers a lot of behaviors. And so I guess saying that a cell can kind of monitor its own stress level is not something new, it's not something very new.
And so I think earlier you said that there was like an already known established generic stress response in
Yeah,
yeast,
a
and
universal stress response.
yeah,
But
and-
that is mapped. That is kind of, there is a group of genes, I don't remember, several thousand genes, 20 or something. And they start, they can read inputs from several different kinds of stresses. Yeah, but I mean, people have mapped out what this stress module can respond to. We have not found evidence that this is the one that's being used here, although we looked for it. But again, I have to, there's a caveat again about the measurement. These measurements have not, have been limited. We don't have single cell, we don't have many, this was done a while ago. I would be really interested to look again with modern technologies into this gene expression.